Hello, my name is Finn Hawkins, and I would like to thank the organizers for the opportunity to present today on the topic Derivation of Airway Progenitors for Cellular Therapy and Disease Modeling. I have nothing to disclose. In 2005, Shinya Yamanaka made a remarkable discovery that introduced the world to induced pluripotent stem cells, or iPSCs. He determined that the overexpression of just 4 transcription factors in skin fibroblasts, or subsequently in peripheral blood mononuclear cells, some of those cells could actually be reprogrammed into a cell type that is molecularly and functionally highly similar to embryonic stem cells. The reason this was so important to the field is because embryonic stem cells have the remarkable ability to form all cell types of all organs in our bodies and are, for that reason, a very attractive candidate for regenerative medicine, but for obvious moral and ethical reasons are not a practical solution to those approaches. In one of the most important experiments done in the last several decades, Dr. Yamanaka, to demonstrate the potential of these cells, injected mouse iPS cells into a blastocyst embryo, and he mismatched the color of the mice so that he could track the donor iPS cells. And here are those important mice generated in our center soon after the first description of iPS cells, showing that these mice are actually made from a combination of the donor embryonic stem cells, the host embryonic stem cells, and the donor iPS cells, and as evidenced by their chimeric coat color, but also within all their organs, iPS cells had actually contributed to their organogenesis. And so iPS cells are very important technology because they are genetically identical to the person they're derived from, so patient-specific. They can be expanded in culture to a near limitless supply. They have the capacity to differentiate into any cell type of interest. And as such, they're amongst the leading candidates for cell-based therapy approaches. So how might we make lung epithelial cells when we think about cystic fibrosis and the major site of disease in the airways of these individuals? Well, we follow the roadmap of normal human development, and that is quite a tricky and complicated roadmap, but it can be broken down into its very simplest stages, which include going from the pluripotent stem cells in the blastocyst embryo to the first major step towards lung, which is the derivation of definitive endoderm. Subsequently, that definitive endoderm forms our primitive gut tube, which will be patterned along a proximal distal axis, and in the anterior portion of that gut tube in the foregut, the first few lung cells will be specified and will be identified by the expression of this key transcription factor NKX2-1. Although not specific to the lung, those first few cells in the foregut that express NKX2-1 are very important, as they will go on to give rise to all of the specialized cell types that make up the proximal airways and also the distal alveoli. Because of the time constraints of this talk, I will not spend much time on the actual details of the differentiation protocols, but in this slide, I want to summarize that in the last decade, our field has made significant progress, first in generating early lung progenitors, and subsequently generating increasingly more mature airway and alveolar cells. I want to give credit to some of the key leaders in the field who've contributed seminal papers, including Drs. Snook, Ryan, Spence, and Goto. And when I first joined Darrell Cotton's lab in 2010, it was with the goal of pursuing this technology, and our group has really focused on deriving these protocols also. Today, I'm actually going to spend most of the time talking about our more recent progress in the last year or two. Basal cells are of major interest to us because they are tissue-specific stem cells of the human and mouse airways. Here you can see an image of the human tracheal epithelium. In the luminal compartment here, you can see the fine wispy cilia on top of the multiciliated cells. But along the basement membrane are these wedge-shaped cells, which are the basal cells. Several decades of work have demonstrated that basal cells can extensively self-renew in homeostatic and in response to injury conditions, and can differentiate into all of the specialized cell types that make up the airway epithelium. They thus fulfill the criteria for an adult stem cell or a tissue-specific stem cell because of this capacity for self-renewal and multi-lineage differentiation. These properties actually make them an excellent research tool because they can be expanded in vitro, and they can also be differentiated in air-liquid interface cultures where they similarly form a pseudostratified airway epithelium. And this approach has been essential in disease modeling, especially in cystic fibrosis. So our group, in collaboration with Brian Davis and my mentor, Daryl Cotton, we were interested in trying to derive basal cells from iPS cells. And this overall was compelling in the field of iPSC technology, this idea of being able to actually generate a stable, self-renewing tissue-specific stem cell from iPS cells would overcome many of the hurdles faced by the field. And our pet name for this project in the lab is a stem cell from a stem cell. So I'm just going to review the progress here, and that is that we have developed a differentiation protocol that initially generates airway organoids after about 30 days of culture. These organoids are mostly immature airway epithelial cells, and the key progress in the last year, which is summarized in our cell stem cell paper from earlier this year, is the ability to then mature these cells into a cell type that is molecularly and functionally highly similar to a basal cell. And once this stage is reached, these cells can be maintained in culture for long periods of time, but when placed in the appropriate differentiation conditions, will undergo multilineage differentiation. They express the key basal cell transcription factors, P63 and NKX2-1, as shown here in the bottom left. They also express the basal cell surface marker, NGFR. This is a low magnification view of some of these spheroids, which express P63, as shown here. And we performed a very in-depth characterization, which can be reviewed in the cell stem cell paper. And this is single-cell RNA sequencing to benchmark iPSC-derived basal cells. And we determined that they were most similar to adult basal cells compared to other airway cell types of both the human fetus and the adult airways. This is one of my favorite experiments. So the question here is whether these cells can actually undergo multilineage differentiation. And here we're plating single iPSC-derived basal cells, or IBCs as we refer to them, at a very low density so that a single cell can give rise to a spheroid composed of hundreds of cells. And if we look at one of those spheroids, we can see that indeed the single basal cell has self-renewed to give rise to more P63 positive basal cells, but also undergone multilineage differentiation, as evidenced by the pink secretory cells and the red multiciliated cells. If we plate these cells in the standard air-liquid interface conditions, you can see that they form a lawn of multiciliated cells here in cyan with interspersed secretory cells and adherent mucus on top. This is an en-face view, and we can look sagittally through that air-liquid interface culture and see this pseudostratified appearing morphology with cilia faced towards the lumen and wedge-shaped cells along the basement membrane. And indeed, when we look for our classic markers, we see that this epithelium is indeed actually organized similarly to the in vivo epithelium, with basal cells along the basement membrane and cilia oriented towards the air. And these cultures express the canonical airway markers of ciliated and secretory cells at overall similar levels to primary controls. Remarkably, and one of my favorite things to look at in the lab, is that these cells will actually produce mucus and have coordinated ciliary beating. This is a low magnification view looking into one of these transwell cultures, showing a mucus hurricane, debris and mucus being rotated by coordinated ciliary beating. We want to apply this technology now to cystic fibrosis disease modeling, and this builds on observations made over 30 years ago now, initially by Paul McRae, who noticed that when he cultured lung explants from CF or non-CF individuals, he could induce a swelling in response to forskolin in non-CF explants, but the CF explants did not swell at all. More recently, the Cleavers and Deckers group used this approach with rectal organoids, where CF organoids are stimulated with forskolin and can be treated with drugs to determine drug responsiveness. So we wondered whether iPS cells and the ability to generate from those from any patient or individual of interest might also be used in a similar approach. And indeed, this is a video showing that iPSC-derived airway organoids stimulated with forskolin over the course of 22 hours do indeed significantly swell. And we previously determined that this was a CFTR-dependent response. So our overall workflow here is that for each individual, iPS cells are differentiated in at least triplicate runs into airway epithelial spheroids. They're then treated with forskolin, and we're quantified at baseline and 24 hours later using an automated imaging software. And we're analyzing at least 200 spheroids per individual. And the first question is whether we can actually identify clinically significant drug response using this iPSC-based platform. And here I'm showing you the results of 3 different Delta 508 individuals treated with either forskolin alone or different combinations of CFTR modulators. And the main point I wanted to make here is that there is clearly a significant response in organoids treated with the triple therapy VX770, 661, 445. And there's also some variability in that response. But we were very excited to detect this drug responsiveness in our system. Can this technology be actually used in an electrophysiological, very accurate, detailed assessment of CFTR function, which is really the gold standard in the field at the moment? So here in this experiment, we're differentiating the same 3 iPSC lines from 3 different individuals, and we're sorting out basal cells using NGFR and differentiating those cells in air-liquid interface culture. And here you can see that actually all 3 patient lines underwent differentiation in terms of the levels of expression of classic airway cell types, including ciliated secretory cells, similar levels of CFTR expression compared to primary bronchial epithelial cell controls, and form a beautiful lawn of multiciliated cells with interspersed secretory cells, and also the persistence of basal cells. And so what about the CFTR function in these cells? I'm showing you one example here of a single Delta 508 patient, where in an Ussing chamber, we're treating the cells with either vehicle or with different combinations of CFTR modulators. And you can see that in response to forskolin, the vehicle control has little to no change in short circuit current. In response to individual therapy with VX809 or VX661, we see very, very modest change in CFTR short circuit current. But in response to combination V445, V661, we see a robust increase in CFTR dependent current. And so we're now applying this to the really key hurdle in the field, which is the class 1 mutations. In the last few minutes, I want to talk about some of the most exciting work I think we have done in the last couple of years, and this is testing whether iPSC-derived basal cells can actually regenerate an airway epithelium. This is the tracheal xenograft model. So just to explain, this involves taking a rodent trachea, which is then treated with repeated freeze thaws, which kills all cells in the trachea. The trachea is then surgically implanted subcutaneously in a mouse with both ends interacting or facing into the environment, so there can be airflow through the trachea. And the cells of interest are seeded into that denuded trachea. And so in our case, the experiment is seeding iPSC-derived basal cells and looking 3 weeks later to see if they've been able to regenerate an airway epithelium. And here's the data. So by H&E stain, we were delighted to see that there actually were cells now covering that denuded tracheal membrane, and they appeared pseudostratified, morphologically similar to an airway epithelium, and we could see early evidence of differentiation into ciliated cells. And if we look at our classic markers, we see the basal cells have lined up along the basement membrane. The ciliated cells here in green are oriented luminally. There are also secretory cells, as evidenced by the SCGB1A1, MUC5AC, and MUC5B staining. And these human cells expressed GFP and TD tomatoes, so you can see here that all of the cells that have regenerated the airway epithelium were actually derived from humans. So in summary, we've made significant progress in the last decade in generating airway epithelial cells from human iPSCs. Here we described the derivation of a self-renewing population of basal cells from human iPSCs. Airway epithelial cultures derived from IBCs may be applied to drug development for nonsense CFTR mutations in the future, and that's something we're currently working hard on. And we demonstrated the ability of human IBCs to regenerate a denuded trachea in a tracheal xenograft model. I'd like to especially thank my mentor, Daryl Cotton, our collaborators, Brian Davis and Shingo Suzuki, and especially thank both Mary Lou Bierman in my lab, who's assisted with many of these differentiations, and Andrew Baracalla, junior faculty, who's really led the CF-focused research in my lab. Thank you, and I'll appreciate the opportunity to present today.
I'm grateful for the chance to present as part of this symposium work entitled Gene Correction of iPSCs in Airway Progenitors, and I have no relationships to disclose. What I'd like to start with is a discussion of how we may be able to use induced pluripotent stem cells, iPSCs, derived from individuals with CF to either assay potential modulator response or to eventually utilize such iPSC-derived cells for therapeutic transplantation. And shown here is really the work pioneered by Dr. Yamanaka, in which mutant primary cell sources, in this case either skin or blood, could be used via reprogramming to generate these induced pluripotent stem cells. And these cells either not corrected for their CFTR mutations could be then differentiated into some kind of epithelial tissue for drug screening or predicting in vivo response to modulators, or could be corrected and then be eventually used either for derivation of stem cells for therapeutic transplantation or maybe simply for demonstrating functional correction. And in our initial work published in 2015, we focused primarily on how to achieve gene editing or correction of CFTR mutations in the iPSCs, and simply as a proof of principle, demonstrating restoration of CFTR function. So if we think about how we may apply gene editing for correction of CFTR mutations, just shown above is a schematic of just the mutant CFTR locus with, in this case, mutation in exon 11, for example. One possibility is to target the integration of the CFTR cDNA together with promoter sequences into a safe harbor locus with the idea of providing for targeted cells expression of CFTR message and protein in order to restore function, even though the endogenous locus remains mutant. We focused rather on methodologies that could be used to directly correct CFTR mutations in the endogenous CFTR locus, with the goal, at least, of recapitulating with the corrected genotype the normal level of expression, cell type specific expression, for example, or levels of CFTR protein in the derived cells. So in the one methodology, one seeks to site specifically correct mutations by use of either CRISPR-Cas9 or zinc finger nucleases to induce a double-stranded break, and then introduces the wild type or normal sequences as a donor, with the goal of then correcting specifically the mutation. We and others have successfully used this, for example, for CFTR mutations. The disadvantage of this methodology is that it's quite specific either for a single mutation or very, very small number. And since CF can result from at least 300 causative mutations, as well as there have been up to 2,000 different variants identified that may affect CF, that the likelihood of being able to take these forward for regulatory approval is quite challenging for each mutation. So another methodology to consider is rather one that's somewhat more universal, in which one targets into the locus essentially a restorative wild-type sequences, in this case, cDNA exons 2 through 27, preceded by a splice acceptor, terminated with polydenylation sequence, with the idea of the transcription proceeding as normal through exon 1, and then the transcription, and then the appropriate splicing, one would hope, to incorporate the corrective sequences. So this is a methodology that in principle is capable of providing correction for any mutant genotypes that lie downstream of this integration. And we've in fact been exploring its use in different introns thus far. Other groups have directly targeted exon 1, for example. So there's just in terms of our lab and collaborators now have demonstration of these kinds of methodologies working either in iPSCs or in primary airway basal cells. And I'll be talking more about this in a talk in another symposium. So if you're interested, I'll direct your attention there. I'd like to focus in this talk primarily on how we may use either the mutant or the corrected iPSCs to give rise to airway epithelial tissue or to airway stem cells. And this is our initial work where we used methodology that was really pioneered, for example, by Hans Snook's lab and also by Daryl Cotton in terms of deriving cells that within a relatively short time frame already gave evidence of being some commitment to lung. For example, NKX2.1 transcription factor, TP63 is a marker of airway progenitors or basal cells, Cftr expression, for example. And in this case, we knew that this was not a robust or specific induction of only lung epithelium, but we thought that it might be sufficient to demonstrate restoration of Cftr protein and Cftr current. And in fact, that was the case, that if we compare either our mutant iPS lines or those that were corrected through site-specific correction at a single allele, we see the restoration specifically here of the band C Cftr protein. And then when these cultures were then subjected to Ussing analysis by Eric Sorscher's laboratory, we saw again that the mutant line gave rise to no forskolin-induced current, but rather the lines that were corrected at single alleles, in fact, gave rise to current. So this was a proof of principle that one could, in fact, edit and correct iPSCs and could then read out at some level the restored protein and function. So as we then began to think about how we may sort of utilize the iPSC technology, we had to evaluate what type of cell, what kind of, for example, lung cell or airway cell could we think about modifying so that it could be used either therapeutically, or we may be able to use those cells unmodified for generation of airway epithelium in culture. And for that work, we really sort of exploited studies that had been done primarily in the mouse, in which it was demonstrated that basal cells of the proximal airway had this potential both for self-renewing division as well as the ability to differentiate into other cell types present in the proximal airway epithelium. And here you see the basal cells here at the basolateral surface. Here you see the derived secretory cells, ciliated cells, and ionocytes. So the thought was, well, rather than just trying to generate a tissue, let's first generate from either the mutant iPSCs or from the corrected, specifically airway basal cells. Let's try to derive stem cells so that they can, particularly in the case of transplantation, they could, when taking up residence, both be able to remain long-term through self-renewing division or through differentiation into the progeny cells. And when we think about how this could be used, if this is the CF airway, with the dehydrated mucus and the inability to expel pathogens, for example, the idea was if we could take cells from the patient, generate iPSCs, generate airway basal cells that were corrected, maybe what we could do is deliver those specifically in some way to this airway epithelium, enable them to take up residence with the idea of contributing then to the differentiated progeny, which would now be Cftr expressing because of the correction back already in the iPSCs. So that's the overall model. Now, in order to do this, we really linked up closely with Daryl Cotton and Finn Hawkins at Boston University, began to develop a number of reporter lines that would incorporate specific fluorescent proteins that we could utilize to optimize the directed differentiation protocol to ultimately give rise to these airway basal cells. And we first started with NKX 2.1 to generate lung progenitors, then marked TP63 to give rise to airway progenitors. TP63 is expressed both in the airway progenitors and then the basal cells. And then we would use a surface marker, NGFR, to identify the cells as they matured into functional basal cells. And essentially, I'm just going to briefly go through this work here where we were able to induce first NKX 2.1, shown here by the appearance of GFP, by taking those cells and sorting them and putting them in what we call airway media, where we essentially remove Wnt signaling. We're able to induce airway organoids that now had a population of cells that were double positive, both for NKX 2.1 and for P63. And these airway organoids could be expanded for very robustly with various passages to increase the number of those cells. What we found was that in that airway media, that although we had double positive cells, we did not see induction at any significant level of the NGFR, the nerve growth factor receptor, but only when we selected media together with supplements, we were able to see now also the co-induction of NGFR. So this was encouraging to us that we may be generating what we call iPS-derived basal cells, or IBCs, and we wanted to see how do they compare, if you will, with primary airway basal cells. For that, we looked at transcriptomics, a number of different differentiation approaches, as well as in vivo engraftability. This is just showing here a UMAP of single-cell RNA-seq data, showing the original airway media-derived cells, and here in turquoise, those from basal cell media. And you can see, as expected, the induction of the NGFR, specifically in this population of cells. These cells also lined up with a basal cell signature. And importantly, when we evaluated these cells here for their comparison with different types of cells from the lung at different stages of development, we found that they corresponded most closely to adult basal cells. Since that time, what we have done is to use recent information obtained by several groups. Here I'm showing the data from Gianni Carrero and Barry Stripp, where they looked at freshly isolated epithelial cells of the lung, and they showed, perhaps not unexpectedly, that there's not a homogeneous identity of basal cells, but rather different clusters that appear through single-cell RNA-seq. In addition to the canonical multipotential basal cells, there's also signatures for secretory primed basal cells, activated basal cells, proliferating basal cells, for example. So what we wanted to do is look specifically whether this signature for the multipotential basal cells could also be identified in our basal cell media derived IBCs. And in fact, you see a good representation of this particular signature in cell types derived in that media. There's also some percentage of cells that exhibit this secretory primed basal cell as well. So I think this indicates that we're at least have a population of cells within those iBCs with this multipotential capability. That capability was also sort of read out, if you will, in the ability to differentiate in air-liquid interface culture, where we saw secretory cells and we saw ciliated cells as well. Now, the idea all along was only to use these reporters to sort of guide the development of protocols that we could eventually apply to non-reporter iPSCs. And just shown here is the incorporation of specific surface markers that we used for sorting at different stages. And in fact, for a variety of non-reporter, either CF iPS lines or non-CF, in the open bars, you can see that some of them give rise to good generation of what we call NGFR positive, EpCAM positive cells, characterized as our iBCs, and essentially other lines that performed rather poorly. So in order to come up with a methodology that we could use more generally, we incorporated a sort suggested by Jason Spence's work, where we sort specifically for F3 EGFR positive cells that enriches for NKX 2.1 positive cells at day 30, and then we get much more robust production of these NGFR positive, EpCAM positive cells, and that's just shown in the dark bars here. When we take the cells generated with this particular methodology, put them into air-liquid interface culture, we see robust multipotential differentiation, well-differentiated airway epithelium that performs very, very well in these Ussing chamber assays. In this case, evaluating, for example, N1303K W1282X iPSCs, differentiating this way for their response, for example, to triple combination therapy. And this methodology gives rise to quite consistent experiment to experiment and clone to clone Ussing chamber measurements. So we have several of our, either from our laboratory or from collaborators, presenting work at this meeting utilizing this particular assay. So I would direct your attention to that if you're interested. So also these iBCs generated in that basal cell media give rise through tracheal xenografts to robust epithelial development as well. So that's promising with respect to eventual therapeutic transplantation, perhaps. So let me just focus now on a few of the sort of differences that we think are important to explore, areas in which these iBC-derived alley cultures do not exactly recapitulate that of alley cultures derived from primary airway basal cells. For example, these cultures here are only stable for approximately 2 to 3 weeks, whereas for airway basal cells, they can be maintained for numerous weeks, several months, for example, and still retain their structure as well as their Ussing readouts. In the case of the IBC alley cultures, we see reduced sodium channel activity relative to that seen in airway basal cell derived alley cultures. And what I like to then focus on is really this issue of development of ionocytes. And what we have seen is that either by single cell RNA-seq or in staining specifically for FOXI1 positive cells, we see a very low to negligible presence of cells characteristic of ionocytes, far less than that seen in the primary basal cell derived alley culture. So in our center, an assistant professor, Sarah Wang, together with Shingo Suzuki, have really been revisiting this whole issue of how best to derive induced basal cells of increased competence, that is, able to give rise to even some of these rarer cell types. And their work is coming along very well. This just shows an increased frequency of ionocytes using their protocols under development, a robust ciliary motion as well. So hopefully we'll be able to tell you about that work in the very near future. I'd like to just acknowledge people in my laboratory as well as fantastic collaborations we've had, which have allowed us to both develop these protocols for deriving airway basal cells and airway epithelium, to test them in different functional ways, either for assaying modulation by different small molecule drugs, or by actually doing genetic correction to hopefully develop cells that can be used for transplantation. And I want to thank you for your interest and for your attention.
Good afternoon. My name is Amy Ryan. I'm Assistant Professor of Medicine at the University of Southern California. And today my presentation is going to be the derivation of pluripotent cells from other species for modeling of cystic fibrosis, in particular ferret-induced pluripotent stem cells. I have no disclosures. So let's start by recapping what is a pluripotent stem cell. So there are two forms of pluripotent stem cells. Those derived from the inner cell mass of a blastocyst, known as embryonic stem cells, and induced pluripotent stem cells, which are derived from somatic cells from a patient or donor and reprogrammed in vitro using the reprogramming factors OCT4, KLF4, c-MEC, and SOX2. These generate these tightly compact colonies of stem cells, which have the key properties of infinite self-renewal and multilineage differentiation. So what are the advantages of using pluripotent stem cells for lung disease? Well, when we're thinking of curing cystic fibrosis, one of the options is cell therapy and tissue regeneration. And with this, we can take patient cells, we can take those somatic cells and reprogram them to pluripotency, generate an unlimited source of matched cells. The advantage of this is that we can differentiate these into multiple cell types from a single donor. And we can also genetically manipulate them using technologies such as CRISPR to correct mutations. We can then clonally expand these, differentiate them, and test out the feasibility of autologous cell therapy for that patient and curing that patient. We can also develop patient-specific models for lung disease by taking those differentiated cells and using those in mechanistic research, and also developing reproducible and adaptable high-throughput screening technology that can be used in precision medicine approaches for developing therapeutics for the patients. So we have human-induced pluripotent stem cells that we've already got these differentiation protocols being established, and pluripotency is already established. So why do we need non-human induced pluripotent stem cells, and why are they useful for cystic fibrosis? Well, the problem is we have no in vivo preclinical model for the evaluation of autologous cell and gene therapy in humans. We have the mouse model and as an animal model, a small animal model, but these really aren't adequate when we're trying to evaluate long-term effects of cell replacements in the conducting airways. And when we're thinking of cystic fibrosis, the mouse model poorly recapitulates the CF lung phenotypes when compared to the human. We know that the pig and the ferret are potentially really good models for cystic fibrosis, and therefore potentially the best model for developing preclinical therapeutic approaches and preclinical models due to the morphological and functional similarity to humans. However, when we take these animal models of cystic fibrosis and look at pluripotent cells, it appears they're not all created equal. So let's take the mouse. We know the mouse as a model of cystic fibrosis, and we also know that induced pluripotent stem cells and embryonic stem cells have been generated, characterized, and maintain pluripotency beautifully in vitro. However, the mouse models are somewhat limited. If we go to a little bit more complex, larger animals such as the rabbit, where we have abnormal phenotypes that are detected in surviving rabbits after a year, induced pluripotent stem cells and embryonic stem cells have been generated, but they're challenging to generate and maintain. Similarly, so are pig and ferret pluripotent stem cells. We have specifically worked with a ferret in our lab. These animal models more closely reflect the pathophysiology of cystic fibrosis lung disease. However, iPSC have been generated and characterized, but they do not maintain well in vitro. And for both of these species, embryonic stem cells have yet to be fully established. What is interesting is that these species actually are very closely related to other species that have also been challenging to generate pluripotency pluripotent stem cells in vitro. And these include canines, felines, cattle, sheep, and equine in the carnivores and the ungulates proportion of this phylogenetic tree. So what is it that makes these animals different? Well, the answer is we don't really know why they don't reprogram so well. But we have focused specifically on the ferret pluripotency, and with our goal for generating a preclinical ferret model for cell therapy. And we've been working with Dr. Parekh at the University of Iowa, who developed an orthotopic transplant model in the ferret for a lung transplantation. And this has the advantage that we can take ferrets, somatic cells, reprogram them to pluripotency, and directly differentiate them into lung progenitor cells that can then be used to engraft either in the donor lung or directly back into the ferret, providing a great model system for evaluating cellular therapeutics. The bonus here as well is that we can go and use CRISPR to correct the Cftr gene mutation, expand gene-corrected lung progenitor cells, and go through the same process of evaluating autologous cell therapy. Problem is we didn't have any induced pluripotent stem cells from ferrets, so that is what we embarked on a few years ago. We took ferret fetal fibroblasts from ferret, we applied the reprogramming factors, and we used human reprogramming factors as has been done for a number of other species, with the goal of generating induced pluripotent stem cells. But we really didn't know what to expect. We didn't know whether they would reprogram and become more like mouse pluripotent stem cells, which are naive and lift dependent. They generate these 3D dome-shaped colonies, or whether they would be more like human primed embryonic stem cells, which generate these more flat and FGF2 dependent colonies. We tried several techniques of reprogramming, but eventually landed upon the episomal reprogramming as the most efficient method for generating ferret-induced pluripotent stem cells. And following a standard protocol that the manufacturer of the episomal cytotune reagents, we were able to generate these flatter, larger, primed and FGF-dependent colonies that here you can see stained for alkaline phosphatase. We picked and expanded these colonies and then went through the standard characterization of pluripotency, including the expression of pluripotent genes such as OCT4, SOX2, and LIN28. This is comparing human pluripotent stem cells and ferret pluripotent stem cells. And you can see here these colonies are very similar and express the same genes. However, the ferret, we also noted that over time, the colonies become challenging to maintain. And although still expressing pluripotency genes, you can see that OCT4 expression is changing and becoming weaker, and the colonies are losing this nice tight boundary, and the cells are starting to migrate out of the colonies. Again, here you can still see another morphology of ferret pluripotent stem cells. These are a little bit more dome-shaped and still expressing the pluripotency genes. However, when we transfer these into lift media to see if they're able to maintain as naive stem cells, they differentiate rapidly. The first thing to do was really compare our pluripotency, or to evaluate whether we were seeing an upregulation of a pluripotent gene signature in our ferret pluripotent stem cells. And when we compared our pluripotent stem cell lines to the original fibroblasts and mature lung epithelium, we can see that they are transcriptomically distinct from the differentiated cells. When we then went in to look at a panel of pluripotency-associated genes, you can again see that the ferret-induced pluripotent lines have upregulation of these genes, whereas the ferret fetal fibroblasts have very low expression of these genes and higher expression of differentiated genes. And when we look, you can see that the pluripotent cells cluster away from both the differentiated lung cells and the differentiated fibroblasts. So this was a good indication we had successful reprogramming. We then wanted to compare the pluripotent stem cells we generated from the ferret to human and porcine pluripotent stem cells by using published data sets for those other stem cell lines. And here you can see, excuse me, gene expression for the ferret on the left, the humans is in the red, the porcine and the pig is in the green here. And then we also compare to mouse, which are those naive stem cells, which are on the right-hand side. And you can see that all three of the ferret, human, and pigs cluster away from the mouse, indicating that these are primed and these are naive. However, you can see that the ferret has the most similarity with the human iPSCs, whereas the pig seems to have some genes that represent mouse and some genes that are more akin to human pluripotency. When you look at how well these cells cluster together, the mouse cluster far away, the human and the ferret cells cluster together and away from the pig pluripotent stem cells. Similarly, when you look at pluripotency-associated genes, you can see that these are most highly expressed in the human, with some of the ferret being as highly expressed as the human and others being much lower expressed. And the pigs have much lower expression of the majority of these pluripotency genes compared to human and ferrets. When we then look at pathways that are upregulated in the ferret pluripotent stem cells, we can see many of these are associated with pluripotency, including OCT4 pluripotency, MAP kinase signaling, nanopluripotency, Wnt beta-catenin signaling, again suggesting that we have activated a pluripotent state in our ferret pluripotent stem cells that is similar to that in humans. Key feature of pluripotency is that the cells are able to differentiate into all lineages. So we generated embryoid bodies from our ferret pluripotent stem cells and looked at spontaneous differentiation into ectoderm, mesoderm, and endoderm. And you can see here efficient differentiation of all those lineages. We also directed the differentiation using combinations of growth factors and cytokines, and again could generate PAX6 expressing ectoderm, smooth muscle actin expressing mesoderm, and SOX17 FOXA2 expressing definitive endoderm. We also did in vivo multilineage differentiation by way of teratomas by injecting cells under the skin. You can see here the teratomas and H&E staining, where we can see cells representing each of the different germ layers, including mesoderm, endoderm, and ectoderm. Again, on the right-hand side, we've stained these sections with markers such as TUJ1 OTX for ectoderm, smooth muscle actin for mesoderm, and FOXA2 and SOX17 for endoderm. You can see here nicely we're getting a secretory epithelium differentiating, representing endoderm, which is what we would like to get for evaluating autologous cell therapy in the lab. So this is great. However, ferret pluripotency or ferret pluripotent stem cells have their limitations. And the major one of these is that they're very challenging to maintain in the undifferentiated pluripotent state. And this is a similar fate that is observed when trying to maintain pig iPSCs, canine iPSCs, feline iPSCs, and equine, to name a few. What you can see here is that we still have colonies forming, but the colonies are no longer tightly bound circular colonies. They're becoming more diffuse with differentiating cells between the colonies. Christiana Sanger in my lab has now developed a technique where we're able to selectively take the pluripotent components of the colonies and split those selectively using a two-phase acutase split. And you can see here that we're able to maintain now slightly nicer, more round colonies, and these colonies stain very nicely for the pluripotency genes OCT4, SOX2, LUN28. Despite this, they're still very challenging to maintain, and when you differentiate them, they're heavily skewed towards ectoderm. This here is a differentiation we did where we're pushing the cells towards definitive endoderm, and although you can see a regulation of FOXA2 and SOX17, you can also see significant expression of TUJ1. When we then take these cells and push them further and further towards lung progenitors, we can see the efficiency of NKX 2.1 induction is very low compared to the human on the bottom. We see just a small cluster of cells able to be differentiated to lung progenitors. This is going to be extremely limiting moving forward when we want to move towards evaluating cell therapy and in a preclinical model, because we're going to want to generate a vast number of cells. So we've worked on improving reprogramming and improving differentiation. First thing we looked at was the epigenetic status of the cells. So we tried a number of epigenetic regulators. As has been shown before, back in 2008, in one of the first papers, showed that if you use the DNA methyltransferase inhibitor, you could both increase the number of colonies and the size of the colonies during reprogramming. We tried this with the ferret, and we can also observe an increase in the number of colonies and the size of colonies using RG108, which is the DNA methyltransferase inhibitor. However, while this improved the efficiency of generating pluripotent stem cells, it did not impact our ability to maintain them as we expand them. One thing we did note, though, was that the epigenetic regulation did improve the differentiation to endoderm. So we have vehicle here on the top and 5-azacitidine here, which is a DNA methyltransferase inhibitor. And when you compare the efficiency, you can see that there is more cells differentiating to FOXA2-SOX17 expressing definitive endoderm. And again, we can see an upregulation of the genes with the 5-azacitidine indicative of a better efficiency of generating definitive endoderm. However, this was still not efficient enough to get sufficient cells moving towards the NKX2.1 expressing lung progenitors. The final limitation of studying ferret pluripotent stem cells, and indeed the other species, is that we have a lack of species-specific reagents, such as growth factors for maintaining and differentiating the stem cells. The genes for reprogramming, mostly we're using human genes rather than cloning the species-specific genes. And we have a lack of validated antibodies for evaluating the efficiency of differentiating and for flow sorting cells. One of the biggest limitations here for generating the reprogramming constructs in our case is that the ferret genome is incompletely annotated, and especially when it comes to the pluripotency genes. But one thing we have done to move forward with this is we've collaborated with Jin-Sen Sun of the University of Iowa, and he has generated an OCT4 reporter ferret line so that we can try and study the factors that are regulating pluripotent state in both embryonic stem cells and induced pluripotent stem cells from these ferrets. So he looks at the which gene to tackle first, and we decided that OCT4, because the 3' UTR is highly conserved across most of the species, as can be seen here by the green highlighting the conservation of the sequence across cats, dogs, humans, and mice. What he did was he generated an iris GFP, and he CRISPRed this into the 3' end of OCT4, and generated a single ferret clone, a ferret that has had successful homologous recombination, included the iris GFP, as can be seen here on the sequence validating it here. He's now expanding these ferrets, breeding them and generating the F1s so that we can look at both embryonic stem cells and generate induced pluripotent stem cells, and hopefully do some high throughput screens to look at factors that may be regulating pluripotency differently in ferrets compared to what we're more familiar with in the humans and mice pluripotent stem cells. So in conclusion, today I've shown you that reprogramming to pluripotency is not equal across all species. I gave you the example of the ferret, but it's this difficulty seems to be common across ungulates and carnivores in general. Ferret iPSC do spontaneously differentiate, and these are heavily skewed towards ectoderm, which makes moving towards endodermal lineages such as the lung more challenging. But the ferret pluripotent stem cells really do offer the opportunity to create a large animal preclinical model that we can evaluate autologous cell therapy, which makes pursuing the standardization of pluripotency in ferrets extremely important. And we hope to be able to improve the availability of validated reagents for the ferrets and move towards a fully annotated ferret genome, which would hopefully significantly enhance our ability to generate good pluripotent stem cells and differentiate them efficiently towards the area epithelial stem cells that we would need to evaluate autologous cell therapy. So with that, I would just like to thank Christiana Singer from my lab, who's done most of the work on this, and our collaborators at Iowa, Jin-Sen Sun and Cal Perec. Thank you very much for listening.
Okay, thank you very much to all three speakers. These were outstanding talks. I think everybody can see the questions in the chat, but I will rephrase them. So first of all, a question for Finn and Brian. Can iPSCs be shared with other cystic fibrosis researchers? And if so, would someone have the experience with human BEs be successful with, would someone who has experience with human BCs be successful with the iPSC cultures? Maybe Finn?
Sure. Yeah, thank you. Be curious to hear Brian's perspective as well. I think the short answer is yes, we've been generating these cells from many different individuals, many different patients with CF and other genetic diseases. And I think our long-term goal is to have freezers full of these valuable cells that are ready to share. We have already been sharing these cells with other investigators. And I think the CFF lab, led by Junji Lee and Jed Mahoney, have been able to reproduce these protocols and have similar goals of having banks of these cells for investigators, I think with a particular interest in the CF patients with nonsense mutations, for whom it's really can be quite challenging to get primary cells for in vitro studies. There are some nuances in terms of it is quite complicated cell culture. These are not the easiest protocols to work with, as the people on the screen are experts. We have established protocols to do so, so I feel pretty confident that with some guidance and with our protocols, we could get somebody up to speed who has experience in differentiating primary cells in ALI culture. The key difference I would say that I should mention in terms of primary culture is that these ibasal cells that we generate in our protocol are maintained in a three-dimensional sphere in Matrigel, so different to how the primary cells are maintained and expanded. And in that format, we don't maintain a 100% pure population of basal cells. So over time, there is some differentiation to secretory cells, which means for our best ALI cultures, we actually do an NGFR sort so that the starting cells for the ALI cultures are actually a pure basal cell population again. Hopefully that's my perspective.
Brian, your perspective?
Yes, and maybe I can add to that a couple of things. The first is, I believe, if I'm not mistaken, is that the CFF laboratory is already establishing a bank of the induced basal cells from specific genotypes. So you may want, I don't know at this point how extensive that is, or but I know that they've indicated that at least some of those would be made available to inquiring investigators. So I would suggest maybe getting in contact with Jed Mahoney, who gave one of the presentations as part of the plenary yesterday. The other thing that I would say is that the one difference that I mentioned at the end of the talk was one has to be a little bit careful with the use of the airway, the air-liquid interface cultures for doing sort of the ussing experiments. That using the protocol that we've published, I think we and other groups have found that they sort of collapse after about 2 or 3 weeks in the alley culture. So one has to just be a little bit more careful in terms of when you hand them off for the electrophysiology as compared with the primary airway derived cultures.
So interestingly, we can generate something that looks very much like the cells you guys have. The protocol is slightly different, but the hurdle we're facing is exactly what you're saying, Brian. We have nice p63 positive cell, they differentiate, and Jed did that for us actually, to ciliated cells and to secretory cells, but we never get a completely confluent ALI that you can use in an ussing experiment. And we're at but we can keep them going though. But they never get completely confluent, and it's so we still think that they are not fully mature. We don't know what the problem is, and we should actually compare with the cells that you have.
Yes, I think that.
Yeah, definitely.
Go ahead, Amy. Sorry.
I was just going to say that's definitely an interesting point. I think another important thing to remember is that
In most cases, we're not yet able to expand these as well as primary basal cells in the 2D standard culture conditions either. It's specifically the three-dimensional expansion that is leading to the more robust ALI cultures. So there's definitely some differences between the iBasals we're all generating that we don't yet fully understand what they are.
Yeah. Okay, another question for Finn from Junji Liu. If you could isolate the basal cell, he asked if you could isolate the basal cell population from the xenograft, and are these then more similar to iBCs or to human primary basal cells?
Yeah, thanks for the question, Junji. This is, I think, a nice follow-up from the last discussion. In our tracheal xenografts that were performed by Brian and Shingo, we did get this nice mucociliary airway epithelium, and one of the interesting observations was that the basal cells had what seemed to be a higher level of keratin 5 expression by immunostaining. And so we've identified now by a number of different analyses that although our iPSC-derived basal cells express a panoply of basal cell markers, the keratin 5 is lower than would be expected based on primary basal cells. So we also wonder whether in the tracheal xenograft that's something that we're missing in vitro to get these cells more mature maybe, is missing and is being supplied in that somewhat in vivo environment. I don't have an answer yet, but that's an experiment we would definitely like to do.
Okay, a question that actually relates to this from Calvin Cotton. Do you have ion transport data from the IBC xenograft experiments, which I think is not trivial to do, but
yes, we don't, but that would also be something we're really interested in doing for a number of reasons. I think, Brian, keen to hear what you think, but what I'm most interested in is actually the lack of ENAC that we see in the iPSC-derived cultures is difficult to explain and difficult to address experimentally. So I wonder if in a more physiologic environment, possibly that is improved. But Brian, we haven't done it, is the short answer yet. But what do you think?
I think it's a great question. With the collaboration we've been doing with Eric Sorscher and Andres Robb, thus far, everything that we sent off has been basically, ALI cultures. But one of the things that Dr. Suzuki is testing with Dr. Andres Robb is we'd like to be able to take these xenografts and actually be able to access the epithelial tissue in it, that it can be used for ussing. So I know they have experiments planned to be able to look at that. We're particularly interested because we would like to be able to do experiments testing. The protocol that Finn and I described are not the only protocols for deriving IBCs. Amy has described protocol, and Jason Spence and Hans had reports on IBCs as well. So one of the things that we'd like to do is to do sort of comparative competition experiments to see how do these cells derived from various protocols compare in their ability to give rise to airway epithelium, restoration of Cftr function. So, Cal, the question that you're asking is a really important one, and it's one that we hope to be able to address. There was some initial work from John Engelhardt's lab where they reported the ability, I believe, to do potential difference measurements directly on tracheal xenografts, sort of still in vivo in the animal, and actually able to analyze that over time. So that's another possible way, is to, in a non-destructive way, being able to analyze, for example, the consequences of gene editing methodologies, for example.
Okay, lots of questions coming and all very interesting. So, but if I can interject one thing, Brian, you said something really interesting. There's at least 3 or 4 or 5 labs that make IBCs through related, but still quite different approaches, in particular with the terminal stages. We all get P63 positive cells, NKX2.1, NGFR positive, KRT5, integrin beta 4 positive. We all get that, right? Yet all our cells seem to be behaving slightly different, or maybe not. Maybe this is the way we assay them. And in fact, there's an incredible need in this field, I think, that all of us get together and, as you said, compare ourselves at the single cell RNA-seq level. The competition experiment and tracheal explants, I think, is very interesting. And I think we should do, it's very important for the field to do that. In this respect, another question and a remark from Martin Minson from the CFFT lab. He said, yes, it's the goal of the CFFT lab to distribute iPSCs to the community for people to try and adapt them as a system for rare genotypes. There's plenty of cells available right now, which is very good to know. And then Scott Randell has a few questions and remarks. First of all, he says he enjoyed the talks very well. An interesting comment for discussion more than a specific question. And Brian and I talked about this right before we started this meeting. Yeah. The supply chain problem with Matrigel is problematic. And yeah, so, and I can confirm that. So we ordered a bulk order of Matrigel, and delivery is promised for April or May 2022. Imagine. I mean, that shuts down a lab that relies on Matrigel. So that's one issue that we have to address and use other reagents. I would also add to this as a personal note. We try to do our cultures all serum free, right? We're all very strict in defining the media. We're all very rigorous about it. Of course, Matrigel is a very undefined media. And I think the field, this is another thing that the field should get together on to get rid of, to overcome this Matrigel issue. And then the second question that Scott had, how will we address the fitness issue so well put by Brian? Brian and Finn, feel free to answer. And then there's another question from Scott, but answer this first.
Brian,
do you want to give it a try, Finn?
After you.
So I think this ultimately, I mean, when one considers fitness, I think one has to qualify is fitness for what? That is, you know, a lot of the work that we're putting the cells to now, really analyzing what is the fitness for those cells to be able to give rise to alleles, or let's say the type of swelling organoid studies that Finn showed. So that's one level of fitness, and we already know at that level that that there are some real differences. For example, the absence of ionocytes that I described. So I would say that's one level. The second is if we think about using corrected iPSCs for some kind of transplantation, I think that's a case where we need to be able to do competitive experiments in experimental systems that allow us to directly see what is the level of engraftment and regeneration of tissue of our iPSCs compared with those of, let's say, human primary airway epithelial cells of low passage. And that's an area that a number of groups are really trying to make headway on, trying to find out what is a good, you know, other than the tracheal xenograft model, what is a good model that we can use for inducing some kind of transient injury and then delivering the cell. So I think I don't have any more to address than that. What is your thinking, Finn?
I mean, I completely agree with you, Brian. I think it really depends on what your goal is for these cells. And so I don't have much to add except maybe a few comments that I'm very optimistic. So while our iPSC-derived cultures are not as durable as primary cells, we do occasionally generate fantastic ALI cultures that will last for 3 months. And so, you know, I think sometimes we're just missing something in some of our differentiations. We have noticed that in those ALI cultures after 2 or 3 weeks where, you know, with the primary cells, you would expect to see an abundance of basal cells that just keep replenishing that airway epithelium. We do see in our iPSC-derived ALI cultures not as many basal cells. So one of our current theories is that our basal cells do not maintain as well once they also start to differentiate. And then the Matrigel is a major headache of ours, and actually we're working hard on trying to improve the durability of our ALI cultures, and have found that by modifying the matrix, actually we get much more durable cultures. That remains preliminary at this point, but we're pretty consistently able to go to 4 weeks or more. But that doesn't get past the issue that Hans raised of the 3D matrigel culture is a problem. It's overly expensive. But I'm optimistic that we can identify improved expansion methods for these ibasal cells.
It's Finn, you say something interesting. So you, in the ALIs, you seem to lose your basal cells, they differentiate and they don't last. We should really put our heads together and the data together. We have basal cells that grow in the ALIs. They never become completely confluent. They make holes, but they stay. Many of them stay as true basal cells with all the markers you need. So we have the opposite problem, and I don't know why that is.
That's interesting.
I'm puzzled. Of course, we generate them in a slightly different way, but I think the field should really talk to each other, because we're all circling the same grail, and we don't have it yet. So another thing from Scott Randall. He says, devil's advocate. Well, Scott, that's nothing new for you. So he says, to accept them as a model, will the FDA require seeing similar effects of Cftr modulators in the iPSC-derived cells as in primary cells from the same donors? What do you guys think? It's a good question. I don't know.
I think maybe Amy wants to address it if she's available, because I believe this is something that she's been working on.
Yes, Amy?
So the level, that was a question on the levels of the CFTR function and what the FDA
so will the
required.
FDA require seeing the same effects of Cftr modulators in the iPSC-derived cells as in primary cells from the same donors, which might not be a trivial thing to do, but.
So yeah, I'm not exactly sure what the FDA are going to require. I would imagine they're going to want to see something of a similar level.
Yeah. Of course, also depends on what you ask for approval for just in vitro studies and validating drugs as an additional validation of a new drug, or, and I wanted to bring this up at the end, engraftment, which, let's be honest, we don't have.
Yeah, okay, I mean, it really will depend on what the goal is. I actually have a comment, just to jump back to that prior discussion, you know, on why the basal cells might be different. I think there's a few things that, you know, all of our protocols somewhat overlook, and is that, you know, in vivo, your cells are differentiating in the presence of all your surrounding niche cells. You've got mesodermal cells around them. You've got immune cells that are interacting. And I was in a session the other day where I presented some of our data where we're looking at macrophage and neutrophil interactions and how that influences basal cell differentiation. So it's not too far removed to believe that, you know, the differentiation of a basal cell from pluripotent state is going to have interacted and maybe been epigenetically manipulated, or, you know, or receiving signals from that environment that we're missing in our culture systems.
I think you make an incredibly good point. Yeah, I agree. Now that we have you, I'll skip one question, I'll come back to the other one, but it's a question specific for you, Amy. You noted a loss of tight pluripotent colonies over time. Have you looked at gene expression changes over the same time frame? And this is a question from Rihanna.
So we did it at the PCR, and we see that, you know, some of the standard pluripotency genes just decrease over time. OCT4, which was specifically one of them. We haven't gone to the extent of doing like single cell RNA seq and looking at maybe what population is differentiating or changing. I'd love to have the funding to be able to do that. We do have a slight limitation still with the ferret, that we haven't got a fully annotated genome, which does have some limitations on missing some of the genes that we're able to track there. But I think that's a good idea, something we would like to do.
Yeah. Then another question from Liwa He. Is ENaC activity, and this is probably a question for Brian, is the ENaC activity decreased due to the lack of gene expression or a lack of function?
It's a great question. We don't have any data yet to answer that. You know, all we know is that when we take cultures either from CF or from non-CF iPS lines, and then you know, establish airway epithelium and then evaluate them first with amelioride exposure, we do not see anywhere near the drop in basal current level that one normally sees for primary. So that's really something that we don't have a handle on at all right now.
I maybe add a comment to that, which is to just extend the observations that in the single cell RNA sequencing of those iPSC-derived ALI cultures, at least at the mRNA level, we're seeing the transcripts for the various ENAC subunits. But the one thing I'm really learning to appreciate is how complex ENAC physiology is. And so we don't know then what's happening past the mRNA level to explain why we see virtually no functional activity of the channel.
Okay, we're out of questions on the chat, but I want to throw in something that we can all discuss. And so we can all make basal cells of some sort, of course, and we can use them for drug modeling and for disease modeling and drug testing and so forth. What are your thoughts on engraftment? Because there's the ex vivo trachea assay, which is very nice. But ultimately, if we ever want stem cell therapies for cystic fibrosis, we have to engraft in airways and, if possible, in small airways. So I know it's a very challenging thing. Any thoughts? I just want to hear what you guys think where this is going to go. Let's maybe start with Finn.
Sure. Thanks for
it's a hard one, I know. I just want to throw it out
there.
No
problem. I mean, my thoughts on engraftment are that as a field, we're just at the very, very beginning of that journey. And it goes without saying it's going to be very difficult. And there are so many different questions and challenges ahead. But we've never been in a better position than we are now to try and take it on. So only a handful of groups have really achieved any type of engraftment in the lung. I think we could spend the entire day debating which cell, which injury model. I learned the other day that the human lung, I think, has 54,000 branching events. So how are we going to target all those branches in CF? But I think there are signs of progress, Hans, and I would be also keen to hear your perspective. So we are actively working on it. We were very encouraged by the xenograft. Why did we choose the xenograft? Was because it's we were surprised that it worked, to be honest with you. It's not truly in vivo. I think we all acknowledge that. And the reason we chose it was because it's so difficult to achieve it in vivo engraftment. And the lung is extremely well designed to prevent engraftment. But we are actively working on it. And I think as we have attempted to pull this off over the years, we've come around to the perspective that if we're ever going to achieve autologous cell-based therapies for the human lung, we're going to have to be able to do it in a mouse first, with autologous cells into a mouse. So I think that's our overall focus, I think, now. And I guess I'll stop there and be very keen to hear what everyone else has to say.
Have you ever tried to engraft the IBCs in an immune deficient mouse with whatever injury model? I think injury models we have in the mouse is one of the problems. The injury models we have are not going to work in humans, and they don't even work in a mouse. I mean, be it sulfur dioxide, polydecanol, naphthalene for the airway, bleomycin for the distal lung is not a good one. So I think that this is hurdle number one. Yeah,
we have started doing that with mouse ibasal cells into syngeneic mice. And we have just started doing it with human ibasal cells into NSG mice. And we have some preliminary data that is not ready to be shared yet because it's too preliminary, but we've just started doing it using a polydocanol injury.
Yeah, we're moved to that too, but it's hard. Brian, what do you think?
Well, I think, I think a lot of the difficulty, this is my own opinion, I think a lot of the difficulty and confusion of the field has been that we're attempting xenotransplantation, that we're trying to take human cells and put them into a mouse background. And I think that we're trying in that context of not knowing whether, and this is all Finn and Amy and I are all part of the CFF-sponsored epithelial stem cell consortium. And this is something that we spend a lot of time talking about how, what is a good engraftment model. And my own sense is that, when we're delivering human cells to an injured mouse trachea, for example, we don't know whether, and if we have difficulty seeing in graft, we don't know whether it's because it's the injury model.
Exactly.
We don't know whether it's because we're delivering the wrong kind of cells, that is in terms of their state of maturation or function, whether they're IBCs or whether they're, expanded airway basal cells, primary cells, or whether it's a mismatch between the two. So we have three fundamental questions that we're all big issues that we're all trying to address simultaneously. And I think the consequences of that have been very little positive movement. So I think this is a case where at least part of the activity has to be a reversion, I think, to testing mouse into mouse. So we can then sort of in an autologous situation, figure out what works, and then begin to sort of move more human into mouse. I'm not sure the only issue is residual immune function in mice that are, attacking human cells. I think it may be just an architectural issue too. So I don't mean to make it more complicated than it is. I just think the problem that we've had thus far has been sort of doing what seemed natural, which is to take the cells we're deriving and put them in. And as a result, there's been little movement forward in the field.
Yeah, I think you're right. In the mouse, the xeno issue, I mean, there's the immunology issue, but that's actually the simple part. You can take very immune deficient mice. So I don't think that's the issue, but it's the mouse environment that may not be very hospitable to human cells. The problem is, of course, ultimately, was that? Yeah, I wanted to go to you, Amy, because I think this is why I took you late, because I think a ferret model would be an amazing model to get closer to human, and we absolutely desperately need these ferret iPS cells. So Amy, I'll let you talk now.
Yeah, I mean, that's what I was going to say. I mean, that was the whole goal of the ferret. We're getting into a larger animal system with closer architecture of the airways and more structure-function relationship to humans. So if we can go ferret-like into like, then we're kind of addressing some of those limitations of going human into mouse or human into another system. We unfortunately have hit the snag that the ferret iPS cells are not like the rhino or other ones that seem to be generated fairly straightforwardly. We have an iPS pluripotency difference as well. So we're going to have to address that first before we're able to get, to that point. So again, I think it is going to be a great system to really be able to address that question once we've overcome that first hurdle of the ferret pluripotency and able to generate sufficient basal cells to be able to use that system. But I think it's one solution that may help us address some of the questions we have.
Yeah, I couldn't agree more. I mean, it's you're absolutely right. I think we're out of question. Does anyone else want to throw anything on the table? Okay, thank you. I think this was a fantastic talks, and it was also a fantastic discussion afterwards. So thank you so much, everybody, for being here and doing that. That's how it moves forward.
Thank you all.
Thank you.
Thank you all. Bye.
Bye-bye.